one that's going to be in the light. And where do you think this one's going to go?
Dark.
The dark. That's right. The dark. Maybe we'll try to get some stuff in here. Look at there. It's a big old apple core. If you were to leave an apple core in your room and you're watching TV and you know yourself, I don't want to get up. The show's really good. I don't want to go to the garbage can and throw it in the bed. What's going to happen to the apple core? It's going to mold. It's going to rot. And what's going to make it rot? Is microorganisms. It could be that mold. It could be fungi. It could be bacteria. It could be whatever. We all have bacteria in our mouth, probably. Okay. Sun is good. Sun is dry. I got to get some carrots. Carrots are hard to poke with. Doesn't grab them. Carrots are good. They're roots. Is it vegetable? Alright, so we've got one that says light and one that says dark. And we're going to need to put one in the light and we're going to need to put one in the dark. So I'm going to let Miss Baxter do that whenever she gets a chance. She can find a place to put one in the light and one in the dark. And if you look at your purple sheet of paper that I've given you, what's that first question say on the front side? What's that first question say? Yeah, what conditions do you think out of those two, light or dark, will make the mold grow the fastest? So you can kind of do that, and that's called making a hypothesis, and that is called an educated guess. Anybody ever heard of a hypothesis before? Okay, that's one of the steps in the scientific method. You probably learned a little bit about that throughout the year. You might have already learned about that. The next thing you're going to do is take down some data. Okay, you're going to do some observation. That's another one of those steps in that scientific method. You're going to look at maybe a day 1, maybe go to day 3, day 4, maybe go to day 6. Every period, if you look at it, take some notes. Look at the one in the sunny spot. Take some notes down. Take some notes down for the one in the dark. And then, even on the back, you can even draw a picture of it if you want to. One in which you can maybe start seeing that mold start to develop. It may take longer than these 8 days, but try to look at it and make sure that you're doing that. And then on the bottom it says, explain what you've learned and write down your conclusions there. Your conclusions should answer that question on the top. You might be right, you might be wrong, but that's why you do a scientific method. And you go through all those steps to see if your hypothesis was correct. And then it says, a last question may lead you into another experiment that you can do at home. It says, what do you think would happen if you kept one of the samples in a refrigerator? And you can kind of make another guess. If you want to do that one at home, you definitely could do so. But that is your mold terrarium, where maybe you will grow some mold. Okay? Thank you guys for listening up so attentively. I enjoyed it. Congratulations to our officers. And hopefully you won't have to bring all kinds of other goodies to the class. So maybe where they want to impeach you or something like that. Kick you out of the job. I think you'll do fine. You look like a fine young man. We look forward to working with each and every one of you. Thank you, Ms. Baxter.